Hello everyone. So today I'm going to talk about movement in and out of the cell. So as we've learned in the videos previously, both eukaryotes and prekaryotes have this cell membrane on the outside of their cells. And so these cell membranes keep all of the organelles in and prevent things from going in and going out, but selectively. So they have a semi-permeable membrane, which means that not everything can go in and out. So in animal cells, this semi-permeable membrane is seen as the phospholipid bilayer, and so this big lovely thing here is just the displayed chemical formula of each phospholipid. And so this is a space-filling example. And when they line up, we get this. These circles are polar ends. And just write that out. And all this inside bit is nonpolar. So this is kind of the same structure as bubbles. Bubbles have that polar exterior and nonpolar layer. And that's how soap cleans grease. So similar construction. So with movement in and out of the cell, we have either active transport or passive transport. And active transport requires proteins and chemicals and energy in order to get larger molecules across the phospholipid bilayer. But there's also passive transport, which is when small molecules can freely cross that phospholipid bilayer without needing any energy. And so one of these methods of passive transport is diffusion, which is defined in the book as the net movement of molecules and ions from a region of their higher concentration to a region of their lower concentration down a concentration gradient as a result of their random movement. So diffusion is not explicitly a biology term, it is also in chemistry and physics. And so one example of diffusion is seen here, where the ink is getting dropped into the beakers, and as time passes, the random movement of the atoms causes the ink to disperse evenly throughout the beaker. And so if we look at this picture, that is more closely following what a few single atoms are doing in the beaker. So they are moving randomly, and it's through this random motion that it spreads out evenly. And again, here is an example of what's happening more at the molecular level. So it's going to start out clumped together, and then as time passes, it will have an even distribution. So it gets a bit more complicated as we put this semi-permeable membrane in. But if we imagine a molecule that can easily pass through the membrane, then this is what would happen, where I have high concentration at the beginning on one side and low on the other, and as time passes it equilibrates so that these are the same concentration at the end. So in a plant, we see that carbon dioxide and oxygen diffuse through the plant cells. So as plants take in carbon dioxide and convert that into oxygen through photosynthesis, both of these molecules are small enough to travel through the different cell walls, and then they leave the plant out of the stoma, which is an opening that allows for gas exchange, so the gases can move more freely. So if we were to imagine a molecule that could not travel across the semipermeable membrane, the system would still want to reach an equilibrium. And so what would happen is, if we say that the space that's not the circles is water, the water would travel across the membrane into that into the higher concentration side right here, and create an even concentration across both sides of the membrane. And so when we are concerned about the movement of water, that is specifically called osmosis. And so the book defines osmosis as the net movement of water molecules from a region of high water potential, which is a dilute solution, to a region of lower water potential, which is a concentrated solution, through a partially permeable membrane. So another way to say partially permeable is semi-permeable, which is a bit more common of a phrase, at least in the US, that I've come across. So again, we see that more clearly that these water molecules are traveling across this semi-permeable membrane to balance out the different concentrations. And so one way that we see this in plants is after we water them. So I'm going to Okay, so I'm not able to click play on this, so I will add it to the assignment in Canvas so we can see what happens after this gets watered. But as we have probably seen, after plants get watered, they go from wilty to nice and happy and upright. And so this, oh, there we go. Quite a lot of water they just put in. So what we're seeing right now is the plant taking in the water by osmosis and distributing it across its various cells.
Water. It's a lifesaver.
So now I want to get into some terms that were a couple of slides ago. So with water potential, most of the time when we talk about concentration, it's talking about the amount of solute per solvent, which is the amount of stuff you have dissolved in a liquid. But when we talk about water potential, we're concerned more about how much water per solution there is. So that means that when we have a low solute concentration, we have a higher water potential because there's more water per solution. And when we have a high solute concentration, we have low water potential. So that means that there's less water per solution. And so then also, as we saw in the video of the plant going from wilted to nice and happy, that is due to turgor pressure, and that pressure is caused from the amount of water inside the plant cell. So if we were to imagine a plant cell with the cell wall here and water inside, we would see, or all the water would be pushing outwards on the cell wall, which helps the plant stand up straight instead of wilting. So plants have specialized cells in their roots called root hair cells, and they are great for absorbing water from the soil because they have a high surface area, and so that allows for more contact with the soil and more area for the diffusion to occur, or osmosis to occur, excuse me. And they also, it says that they have a high high concentration of dissolved ions in their root hair cells. So as we saw before with the semi-permeable membrane in the beaker, if I had a lot of things inside one of the sides, all my water would rush over to try to change that concentration gradient. And so that benefits the plant because then it will help the water go inside the root cell. The concentration of solution that a cell exists in is very important to the condition of the cell. So I'm going to talk about hypotonic, isotonic, and hypertonic solutions. And these are concerned with the concentration concerned with comparing the concentration inside and outside of the cell. So in a hypotonic solution, the concentration is lower outside of the cell than inside of the cell. And so this creates a situation where the water will try to rush into the cell. And in an animal cell, that will cause too much water to rush in and will break the cell membrane, which is also called lysed. But in a plant cell, this is a good thing because it has that thick cell wall that will protect it from bursting. And so it will just increase the pressure of the water on the outside and make it turgid, which is a way to say that it has enough turgor pressure. So in an isotonic solution, this is ideal for plant cells. It will not have any net flow of water across the membrane. And so this is a good homeostasis for the animal cell. But in a plant cell, this is not enough water, and so it would start to limp or go or wilt, or a term here is flaccid. And then in a hypertonic solution, we see that the concentration is higher on the outside. And so that will cause a net flow of water out of the cell and into the solution, which in an animal cell will cause it to shrivel, and in a plant cell it will also cause it to shrivel, but then we see this that the cell membrane is pulling away from the cell wall here, and that means that it has plasmolyzed, and if it's done this too much, it will kill the cell. So the difference here is that in an animal cell will die in a hypotonic solution, and a plant cell will die in a hypertonic solution. And I try to remember the difference between hyper and hypo as, like, if I'm feeling hyper, I have a lot of energy, so then in a hypertonic solution I have a lot of solute. So if we compare diffusion and osmosis, diffusion is the movement of the solute molecule from high to low concentration, and osmosis is concerned more about the solvent molecule moving from high to low concentration. And so, especially in biology, the solvent is water, so we usually say that osmosis is the movement of water across the cell membrane. And so another difference between the two depends on the size of the molecule. So small molecules can diffuse across the membrane, whereas larger ones cannot. And so that's when we get our difference in solvent levels.